Now that you have some background information on cells, we need to talk about the specifics of the parts that cells are made of. We're going to start from the outside and deal with the cell membrane, also known as the plasma membrane. This is simply a border. Anything outside that cell membrane is outside the cell. Anything inside that cell membrane is inside the cell. This is the border in between inside and out. It's a barrier. Okay, it prevents things from getting in. It prevents things from getting out, and yet the key phrase that you need to know about the cell membrane is that it is selectively permeable. Permeable means it lets things through, but this is not just permeable, it's selectively permeable, meaning it lets some things in, it lets some things out, but remember, it's also a barrier. It keeps some things in, and it keeps other things out. You'll see the phrase that comes second here on the screen being fluid mosaic. Those of you who know anything about art know that a mosaic is oftentimes a picture that is made out of tiles, different colored tiles assembled in the correct way, and you step back from it and a picture of something has been created. That's because the cell membrane, as you can see here, is made up of a whole bunch of different pieces. So you've got some pieces that are labeled cholesterol, you've got some pieces labeled as lipids, proteins, sugar chains, which are just carbohydrates. So it's made out of a whole bunch of different little pieces, like a mosaic, and yet it's a fluid mosaic. It's got a fluid quality to it. This protein that you're looking at right here, 10 minutes from now might not be in that same position anymore. It might be over here somewhere. So it's not like a brick wall where you put one brick into place and leave it there and it could be there for the next couple hundred years. Instead, there's a fluid quality to it. A lot going on in this page, but it's all associated with the nucleus. Remember, the nucleus defines a eukaryotic cell. Nucleus is really just a bunch of parts put together here. The nucleus as a whole is made out of, for instance, a nuclear membrane. It sounds like cell membrane, and it is like a cell membrane. It's a border. It's a barrier. It keeps some things inside the nucleus. It prevents things from getting in, and yet it's also selectively permeable. It lets some things that are necessary come and go into and out of the nucleus. It achieves that through what are called the nuclear pores. You can see a bunch of holes over here. Holes are just pores. And so things that need to get into the nucleus or out are going to enter and exit through those pores. One of the main things inside the nucleus is a structure called the nucleolus. You can see it as this dark mass here. And that particular cell part is one that produces another cell part called a ribosome. Lastly, located in the nucleus, and probably the most important thing about the nucleus, is that it contains chromosomes. They represent your blueprint. It tells the cell what that cell is supposed to do. If it's a brain cell, if it's a skin cell, if it's a kidney cell, each of those cells have to have a job or a responsibility. And in large part, it's those chromosomes and the information encoded on those chromosomes that tell that cell how to do its job. And of course, those chromosomes also get passed on from one generation to the next, which is what allows for heredity to take place. The majority of what we're going to talk about from here on out would all be referred to collectively as organelles, or mini organs, and ribosome represents one of those. I mentioned that the nucleolus, located inside the nucleus, produces those ribosomes, and then those ribosomes' job is to make protein. That's it. They make protein. Some of them are free-floating around the nucleus and make protein. Some of those are attached to another structure called the rough endoplasmic reticulum. But all in all, the ribosome's job is simply as little factories that crank out protein necessary for the cell. That ribosome leads naturally into talking about the endoplasmic reticulum because some of the endoplasmic reticulum has those ribosomes embedded in its walls. There are two types of endoplasmic reticulum, rough and smooth, and both are pictured here. The red colored one over here that appears to have a bunch of dots on it, those would be the ribosomes to which I was just referring. They are embedded in the walls, and therefore if you would say run your finger across those, it would be bumpy, and therefore rough, and so thus the name rough endoplasmic reticulum. Notice here the lighter colored portion, there are no bumps, there are no ribosomes attached to there, therefore smooth. In terms of a function, because the ribosomes are embedded in the rough endoplasmic reticulum and they serve as little factories of protein, the rough endoplasmic reticulum is going to take up that protein and it's going to transport it and modify it. The smooth endoplasmic reticulum, not affiliated with any ribosomes, therefore doesn't really have an affiliation with any protein. Instead, its job is to make lipids. When the rough endoplasmic reticulum transports and modifies those proteins that were made by the ribosomes, oftentimes the protein pinches off in a little sac called a vesicle, and you see a vesicle right here. And those vesicles make their way to this particular structure known as the Golgi Apparatus, or also called Golgi bodies. And as it states here, this funny-looking set of sacs acts as what's sort of like a sorting and distribution center. Some of the proteins coming to it from the rough endoplasmic reticulum and elsewhere need to be sorted. Some of them need to be sent, let's say, to the cell membrane. Others need to be exported out of the cell. Others maybe need to be used at a different area within the cell. And so that all has to be organized, and that organization takes place here in the Golgi, and you can see coming out the other side of it more vesicles on their way to whatever their intended destination happens to be. A lysosome is one of those places where some of those proteins might be sent. The lysosome contains what are called hydrolytic enzymes or digestive enzymes. All they do is catalyze reactions that break things down. Sometimes the cell needs to break things down, like, let's say it's a white blood cell and it traps a virus and it wants to destroy that virus. Well, the lysosome could handle that. Let's say a ribosome is just worn out and doesn't do its job anymore. Well, you don't want it just hanging out within the cell. So the cell would want to destroy that and use its parts over again, which it can do if the lysosome breaks it down. The other weird part here, with all the hydrolytic enzymes that are inside the lysosome, is that the cell can perform what is called apoptosis, which basically means it can destroy itself from the inside out. It can crack open its lysosome and release all the hydrolytic enzymes that are in it, and it can destroy itself. It can basically commit self-suicide. Mitochondria, that is the site of cell respiration, which is just a fancy way of saying when the cell needs a source of energy, it's the mitochondria that produces that energy. The cytoskeleton, just like your body has a skeleton and that skeleton determines the shape, so too does each cell. Each cell in your body has a specific shape that's designated based on whatever function that cell has. And the cytoskeleton, composed of microfilaments, which are really small, intermediate filaments, which are a little bit bigger, and then microtubules, which are the biggest, form a latticework throughout the cell that provide the shape and structure of that cell, and even provide a place where some things can move around the cell. Centrioles are a bunch of little microtubules put together in sets of 3 that play a role in cell division that we will talk about extensively later when we get to the idea of mitosis and meiosis. And if we stop at that point, basically what we have is an animal cell. Now remember, we're dealing with eukaryotic cells in general, but now we can break eukaryotic cells down into different types, one of those being what composes our cells, which of course we are animals. And so all the parts that I just mentioned would be necessary for an animal cell.